quick word before this one starts. What you're about to hear is a conversation I've wanted to have for a long time, and in some ways, the conversation that explains everything I've been working toward. I'm a physician, so for years I've worked with families affected by autism, and somewhere along the way I stopped trusting the textbook explanation. The lab data kept showing us something that didn't fit, a pattern, quiet, persistent, the same markers turning up again and again, and it pointed somewhere strange, toward the idea that in some of these children, the body may be producing too much of a naturally occurring psychedelic, a chronic, low-level altered state starting in the gut and shaped by our own genetics. If that sounds far-fetched, I get it. It did it to me too. So I went looking for the one person who'd know whether it was even possible. And I found him in Dr. Stephen Barker, who has studied this exact molecule longer than I have been alive. Before we get into the science, two things I want to be honest about up front. The study that we're doing with UC Davis and Stanford is a hypothesis, not a conclusion. I'm not telling you we've figured out autism, and neither is Steve. What we're doing is asking the questions carefully and holding it to a high standard. And second, even if you've never thought about any of this before, stay with it. Because what Steve has spent 50 years uncovering isn't really about one disorder. It's about what your brain is doing every night while you sleep, where your dreams come from, and a piece of human biology that was hiding in plain sight the whole time. Let him tell it. Here we go. There are scientists who spend a career answering questions, and then there are the rare few who spend a career insisting a question deserves to be asked at all. When the field looks away, when the easy thing is to call it impossible and move on. My guest is one of those few. For nearly half a century, he has chased one of the strangest ideas in all of neuroscience, that the most powerful psychedelic molecule known to humankind is not something foreign we put into our brain, but something our bodies itself may make. In 1976, when this was barely a whisper at the edge of legitimate science, he began the hunt. And in 1978, his dissertation gave the idea a name no one else had the courage to commit to paper: N-dimethyltryptamine, or DMT, an endogenous hallucinogen, and this would define his life's work. And here's what sets him apart. In a field forever intoxicated by its own wonder, he became its conscience. When the literature was 50 years of noise and wishful thinking, he wrote the review that demanded rigor. When others chased the spectacular, he chased the truth, because he understood that the fastest way to destroy an extraordinary idea is to fail to hold it to an extraordinary standard. And the idea survived, because the body does make it. Molecule by molecule, study by study, through his foundational work, through the brain biosynthesis confirmation of 2019, through his landmark review last year, what was once hearsay is now data. He is Professor Emeritus at Louisiana State University. He's the steadiest hand and the sharpest mind this science has ever had, and there's no living person who has carried this field further, longer, or with more integrity. It is my genuine honor to welcome Dr. Stephen Barker. Stephen?
Well, thank you, Christian. I really appreciate the opportunity to discuss particularly the new discoveries about endogenous hallucinogens, and particularly DMT.
Well, for many of us, it was like at one point, DMT, what is that? And I heard about it first with the reports about ayahuasca and then Rick Strassman's work. But maybe let's start from the beginning before we dive into too deep waters. Tell us, Steve, who you are and what sparked your interest in endogenous DMT back in 1976. What was the scientific landscape back then, and what made you think this is worth decades of my life?
Well, in 1976, I was a graduate student at the University of Alabama in Birmingham Department of Psychiatry Neuroscience program. And I had joined that group of scientists and researchers in that laboratory after hearing a lecture by one of its members in 1972. I was taking a course in natural products, and of course we eventually came to a section where we discussed plant hallucinogens, the psychedelics in plant materials. And the professor at that time was Dr. Fred Bennington, who was in the neuroscience program at UAB. And he mentioned, just kind of in passing, that they were initiating studies to try to determine whether or not hallucinogens of the indole class, which dimethyltryptamine is one of the major representatives, were naturally produced in humans, but particularly in patients with a diagnosis of the syndrome of schizophrenia. So I heard that in class and went, what? Yes, I need to be part of that at some point. And one of the reasons for that was I was a very lucid dreamer as a kid. And I had had these dreams that took me to places that I had never seen before, and as dreams will do. And I had on one occasion a dream that became somewhat repetitive over a period of a few years of flying out the window of my bedroom, was actually our dining room in a very small house in West End in Birmingham, and going up above the clouds and soaring over the landscapes that were just fantastic, in color and in sight, and coming to this barren hill with this large Victorian home sitting on the top of it. And I came down to the set of double doors in the front, and there was a spiraling staircase, that went up to a very bright light, right? And I started running the staircase, but the more I ran, the further and further away it got. And, I started, feeling stressed, and I started repeating to myself, I'll never know. I'll never know. And I would wake up from that dream still saying, I'll never know, and crying. Right? It impressed something that may have already existed in me, just curiosity about my desire to understand the world, even at a fairly young age. I was always, as a kid, interested in science, and had questions about about mysticism and myth, and about the events that people claim to have experienced with near death, with the visions and reports that were in different religious texts, that I came to read and try to understand. And of course, the world of dreams. Where was this dream world? What did all that really mean? And in 1969, I had occasion to come upon the psychedelic buffet that was being offered of the times, and decided to have myself a helping, and experienced, psychedelic drugs for the first time. And of course, my experience of that was like, this is very familiar. This is very much like these lucid dreams, and although on occasion they were so bizarre that I knew that it was just something that it was affecting in my brain.
May I ask if that included DMT?
Not at the time.
Okay.
Close relatives, LSD and psilocybin.
Hmm.
But I started studying in that area, and of course that went into chemistry, with the idea that isn't it great to be able to manipulate the matter of the that exists in the world to create, new things. And of course, I was very interested in the idea that uh, was put forward by one of the companies at the time, Better Living Through Chemistry. So, and that, of course, included, drug chemistry, not just psychedelics, but therapeutics as well. That you could actually create something that healed people or improved their life or you know gentled their symptoms. So I also learned about schizophrenia, and of course, during that period, the idea was a splitting of the mind, of the psyche. Of course, that's not really what it is, but things that affected through disease and disorder our ability to properly perceive our environment, what we call this world. And then, of course, what people constituted as the other world, the world of spirits and souls and of dreams. So, natural products chemistry, of course, was part of it. And Dr. Beddington's comments, addressed what I was really interested in. As I said, I really felt I had to go into that area and join their laboratory. Well, it was 3 years after hearing that before I did, right? Because like all graduate students, money became more of an issue, so I had to go to work. And I was at Cal Biochem in San Diego, California for 3 years. And of course, being in California afforded me a few other opportunities. And when I came back, I joined the program and started in that area. There was, of course, some issue with being able to do that research. We were still laboring under the 1970 Drug Act that had been passed in the Nixon administration that made doing such research difficult. But there was still an interest from the military and the CIA and others about whether or not these psychedelics could be used for national security purposes, let's say. So there was some research being done, and some of that research was being done there at UAB. And the people that were involved in it kind of set the stage for an awful lot of this. The head of the program was Dr. John Smithies, and his mentor was Dr. Humphrey Osmond. Now, Humphrey Osmond may be familiar to a lot of people because
Yeah, him and Hoffer. I read Hoffer's book and he
yes,
mentioned him frequently. Yeah.
yeah, Hoffer and Osmond basically started the area of biological psychiatry. The idea that treating mental disorders of all types was to be really examined and put into a position to cure or affect these disorders through identifying the biochemistry that had gone wrong in these individuals.
The orthomolecular science, correct? Is that how they
Yes.
framed it? Yeah.
Yes, they were initially very big on ascorbic acid and nicotinamide,
Yeah. Nice.
taking
Yeah.
massive doses of these vitamin supplements. But, the idea arose, I think, mainly first from Hoffer, uh, but also from Osmond, and Smithies was with him at the time, that perhaps there was an endogenous schizotoxin. There was some compound that was formed only in schizophrenics.
Adrenochrome.
Yes, that altered the neurochemistry of the individual, the patient suffering from these different syndromes and disorders. So they started out, yes, adrenochrome was one of them. There was one related to mescaline. It was DMPEA. It was dimethylphenylethylamine. that was eventually discovered.
Adrenaleutin, was that one?
Yes, adrenaleutin. And the uh, search for those compounds is naturally occurring in psychiatric patients began. And various things came up from time to time. There was one that was thought to be diagnostic in the urine of schizophrenics called the pink spot, right? It was supposedly one of these mescaline-related compound structures, and it was related to DMPEA, right? But further investigation showed that this compound, the pink spot, actually came from the excessive drinking of tea in, by the patients that were on the wards where the research was being carried out.
Oh man, of course.
Right. So the pink spot died. The adrenoleutin, although it was found, it was not found to be particularly psychogenic. And the adrenochrome was also found and was also found to be uh, not hallucinogenic necessarily, but kind of psychotoxic, right? It still would produce delirium and some other things, but not a true hallucinogen. So the stimulation for going after compounds related to mescaline as a possible schizotoxin came from the popularity of mescaline at the time. And my major professor from a PhD, who was Fred Bennington, had come up with an improved synthesis for mescaline in 1951. And when he was approached by Dr. Humphrey Osmond, he gave Humphrey Osmond the mescaline that he gave to Aldous Huxley. And so these people were there at the forefront of the psychedelic revolution. And of course, Humphrey Osmond is the one who coined the term psychedelic, his mind manifesting in his discussions with Aldous Huxley, right? They were sending letters back and forth, and uh, Dr. Osmond liked the term psychedelic, and Huxley liked phenero time as the name for these types of compounds.
Thank God the former survived.
Yeah. So, um, and Dr. Osmond and Dr. Smithies were both there at UAB at the behest of a benefactor in Birmingham who had a sister who suffered um, from schizophrenia. And he had put up the money to establish a chair for Smithies in the university and brought Dr. Osmond to the mental institution that was in Tuscaloosa, Alabama. So they were both there at the same time and were, of course, initiating more research into this area. So that kind of set the stage for my entry into the program to be around some of the people who were there at the beginning of all this and that initiated the search for endogenous psychotogens or schizotoxins that eventually led to the discovery of dimethyltryptamine is naturally occurring in human brain.
Wow. Wow. Very interesting to learn about
Yeah, it was a
the
cool
kind of
time.
the origins and the people who were involved in this. I mean, it's
Yeah,
incredible.
I met Dr. Osmond a number of times, and he would come to visit Smithies, from Tuscaloosa. It's only 40 miles away. And he would sit out in the hallway waiting for Smithies to become available. Very busy man. And being an active psychiatrist and running the program, doing research, Smithies was into just about everything. He let me read his letters from Carl Jung and from Freud. He had letters from Einstein and a number of other people, right? And he would exchange letters with these people discussing a range of topics, like quantum chemistry. He was big into quantum chemistry at the
Wow.
time. And this was in actually occurred during the late 60s, early 70s. So Dr. Osmond, while he was sitting there, every now and then I'd come out of the lab and run into him, and I get to sit down and talk with him. He was a very jocular man,
I mean, do
very,
you think
very
Freud
funny.
and Einstein even maybe
Yeah.
had experience with psychedelic compounds?
Well, I really don't know for sure. I know Freud had taken mescaline,
Hmm.
right? That I'm certain. But and Freud actually believed that there was also some neurochemical foundation for a lot of psychiatric disorders. But
Wow.
he stayed more on the talking psychiatry plane.
So that's not
As Einstein, I'm not so sure.
so that's not as recent as 2019, but really Freud already was somewhat hypothesizing that
Yes.
this could be the
Yeah.
case. That's incredible.
Yeah.
Yeah, and even back then, when he talked about Hoffer, for example, in his book, he talks about how they came to discover the schizotoxicity of adrenochrome because there was this guy who was traveling and he ran out of his asthma medications, and back then it was actually adrenaline that you inhaled, for the bronchodilation.
Hmm.
And the pharmacist says, "I'm so sorry, man, but the only vial that I have is expired." So the adrenaline oxidized into adrenochrome. And he's like,
Right.
"It's so weird. It's pink and it's not the usual color." And so he used it anyway because he had nothing else. And he came home and I think he was completely freaking out. He packed his bags. His wife is like, "What the heck are you doing?" And he was like completely paranoid. And
Hmm.
so how we discover things sometimes is
Yes.
incredible. Um,
Yes.
but that's very interesting that you had these connections. So I'm sure you were really pumped at an early age, like to be in contact with the kind of grandfathers of psychedelic medicine.
Yeah, it was such an amazing opportunity to be in a southern town where these giants of psychiatric medicine were present at UAB, which is, of course, a huge medical center now,
Right.
and be able to work with these folks.
So your early work in 1980 showed how MAO, the monoamine oxidase enzyme, breaks down DMT in the brain.
Mhm.
But the bigger insight was if the body makes it endogenously, it must have a biological function or purpose. What was your initial hypothesis about what that function might be?
Well, in 1976, um, starting in 1974, um, the people that were working in the laboratory with Dr. Smithies were starting to get data on cerebral spinal fluid from schizophrenic patients to compare to normals. So whatever that is, but to control patients. And they were discovering low levels of DMT and another psychedelic called 5-methoxy-DMT that comes from serotonin. Right?
You mean
And
low levels of DMT, not compared to a standard, but just low levels of DMT. They were
just
excited
low
that they got
levels,
it. Okay, okay.
right? Yeah. And the methodology that they were using was not, by modern standards, really all that great, right?
Were the chromatography plates?
Yeah, well, this was on gas chromatography using packed columns, which were, fairly large columns. And the peaks, rather than being nice, sharp peaks, they would come out as kind of like lumps. But they use the same criteria, that it has the same retention time and responds to the detector that they were using. But the detector at the time was not exquisitely sensitive and it was not definitive in terms of identity. So in 76, they published their paper showing that these compounds were present in the cerebral spinal fluid of patients with a diagnosis of schizophrenia. But more importantly, they were also in the cerebral spinal fluid of the control patients. So the idea that this psychedelic was being produced only in psychiatric patients was basically proven wrong. Right? But the exciting thing was, well, what's that doing there? if it's not responsible for some of this. Of course, they weren't really able to measure it quantitatively very well, again, because of the technology. So they improved the technology and made it more sensitive and began detecting more of it. And then finally were able to do, with more modern technology, with instruments they acquired in 1977, that used thin capillary columns that gave sharper peaks, and mass spectrometry, which identifies compounds according to its molecular weight and how the compound breaks apart when bombarded by electrons. And that forms basically a fingerprint of the compound. It's unequivocal identification to a scientific certainty. So they reran some of their samples and collected new samples, and again confirmed that these compounds were being released into the cerebral spinal fluid, and quantitated them. But what they were quantitating was just the diluted sample that constitutes the cerebral spinal fluid. It was not informative as to whether or not DMT specifically was being formed in greater levels in certain brain areas or anything like that, but it was definitely there. So there was sufficient evidence to make the claim that this was a naturally occurring compound in human brain, right? So to begin to really study that, you had to understand the metabolism of the compound, not only the biosynthesis, perhaps storage in brain, its release, and its levels, but also its metabolites, so that you get the whole picture. You'd add all the metabolites together that could have originated from that one compound, and you've got the total production. And if you're going to compare amounts produced between one population and another, you need to know the total amount.
Could
So,
you just in a nutshell walk us through really quick, like just the raw synthesis,
mhm,
precursor to metabolite, which enzymes are involved and
yeah.
what compound, where does it come from?
Well, dimethyltryptamine is derived through biosynthesis from tryptophan, which is a common amino acid in our diet. Tryptophan is in all proteins, and is the highest source for tryptophan is protein. So it gets broken down into the gut and absorbed, goes through the bloodstream to the brain, and is distributed there to form a range of different compounds that we use as neurotransmitters, neurohormones, and neuromodulators, right? And to constitute structure of newly synthesized proteins, right? And other compounds. So tryptophan can be converted to tryptamine, which is tryptophan minus its carboxyl group, right? It's a carboxy amino acid. The carboxy group is removed enzymatically by decarboxylase. We really know how to come up with the names. And, it forms tryptamine. Well, tryptamine can be methylated on the ethylamine side chain um, on the nitrogen to give first one methyl group, an N-dimethyltryptamine, and then the second group, a dimethyltryptamine. So that's where DMT comes from. And the enzyme is supposedly an indole N-methyltransferase. It just transfers methyl groups from methionine, which is another compound we get in our diet and is important in all methylation reactions in the body. And, DMT is formed. But, a lot of the tryptophan goes also to form serotonin. It also forms melatonin. And tryptamine itself is a neuromodulator. It's a trace amine compound that has its own receptors. It's called
And
the
then
TAR.
I think then if you hydroxylate serotonin, you get bufotenin, right? Can you
Yeah,
explain that?
that's the thing. In forming the serotonin, you form 5-hydroxy tryptamine, right? and that's serotonin. But if you take that 5-hydroxy group and you methylate it, again, in a methylation reaction, you get 5-methoxytryptamine, which has its own properties at different receptors. But then you methylate the nitrogen like you do for DMT, and you get 5-methoxy N-dimethyltryptamine, which is 1/10 as potent as LSD. Right? So, the formation of that compound was one of the ones that we searched for, as well as DMT. And we also, did some searches for bufotenin in brain, but less of that was found. Bufotenin is just 5-hydroxy in N-dimethyltryptamine. So if you take serotonin and only dimethylate nitrogen, you get yet another hallucinogen, bufotenin. And that compound is found, however, in urine from humans in fairly large, not fairly large compared to some things, but in quantities that are easily measurable. And a number of studies have shown that. And low levels can be found in the blood. But it appears that it may also be being formed in the gut. And, that's one of the things about pursuing these compounds. We need to start looking in a lot of different areas, but understanding their biochemistry from synthesis to metabolism and being able to quantitate these levels is an important tool in being able to understand where these compounds are coming from and what they're doing. So that's what we set off to do in doing the metabolism studies. So DMT is primarily metabolized to indolacetic acid. Well, that's just cleaving off the nitrogen that is on that side chain along with the methyl groups, right? So there's an enzyme called monoamine oxidase that carries out that step, and it is the major metabolite. There's another metabolite that is DMT N oxide, which is, again, an oxidation step where the N-dimethyl group that's on the side chain is converted to an NO, a nitroxide, type molecule. All right, so it's that molecule is very water soluble, as is indolacetic acid, and both are readily excreted in the urine, get into the blood from from the brain and are excreted in the urine.
I see the indolocinic acetic acid. Is it the IAA
Yes,
or 3-IAA or something?
yes,
I see that.
pre-indolacetic acid.
I see that on some of the functional medicine metabolic testings. I don't know now if it's the organic acid test or it's a different company. But many times we have like no clue. Okay, what's the meaning of this? That this is high or low. But now we know, and at least it can suggest that there might be this process occurring where maybe the regular metabolism of tryptophan is favoring a different pathway. And I know there's the kyurinic pathway, the
Yes.
quinolinic acid, and so you can kind of make some perhaps suggestive conclusions that this could be occurring. But so you would think, okay, everybody should jump on this, and let's say we're in the 1990s now on your career. Why doesn't just everybody measure it? So when you first published your 1981 review, no, well, let me get to that in a second. Back then in the 90s, what were the technical bottlenecks that prevented you from measuring this just, more frequently, endogenous DMT accurately in human samples? How did you, and I wrote down here, Ethan McElhenney, solve those problems?
Yeah, Ethan and I worked on ayahuasca. His dissertation, he was my graduate student. His dissertation was on components of ayahuasca and how they are absorbed, excreted, and metabolized. So in that process, we developed an assay for measuring the N oxide, which it was not been readily or repeatedly being measured in the literature, being reported in the literature. And also indolacetic acid. We had an assay that covered, I think, some 25 different compounds that were the precursors, the compounds themselves, and the metabolites, and reported on that in blood and urine of people that had consumed ayahuasca. That method was developed with a newer extraction procedure that we came up with that isolated these compounds in fairly high percentages from these types of matrices, blood and urine, and that then assayed them using advanced mass spectrometry. That's called, you can call it an MSMS instrument. It's a mass spec. The mass spec measures its own ions in another mass spec to give greater certainty, and allows you to manipulate those ions to do experiments to identify their overall structure. And then we eventually did that on what was known as a high mass, high resolution instrument that measured the exact mass of these compounds as well as the fragment ions to give even further definitive identification. Right, so there could be no question that this was the proper identification of these compounds. Now that some of those methods have been applied by others that are now doing DMT pharmacokinetics studies for the use of DMT as a therapeutic. We demonstrated basically how you could go about this, and using blood or urine samples. And that has allowed some further discoveries related to the normal excretion of DMT. But see, here's the thing: in administering ayahuasca, you're kind of loading the system with DMT. You're putting significant amounts into the body that's far greater than what the brain is perhaps producing, or that may come to the circulatory system from other tissues or other areas. So those are easy to measure. You can find them. But if you look at in the administration of DMT by itself, in the absence of a enzyme inhibitor that might stop that metabolism and thus keep it around for a longer period of time, you still don't see significant levels of DMT in blood, and certainly not in urine. DMT is rapidly cleared, it's rapidly metabolized, rapidly absorbed, rapidly distributed, rapidly metabolized. So a very, very small amount of that DMT administered even shows up that you can measure. So think of what the levels in brain might be and how much of that actually makes it into the circulatory system. Measuring peripheral levels of DMT has proved to be somewhat daunting. You can measure some of the metabolites. I think people have seen some N oxide of DMT, which counts as a DMT molecule. And of course, the interest in being able to measure indolacetic acid as the major metabolite has gained new interest. But one of the complications of that is that indolacetic acid comes from tryptophan from a variety of pathways in the periphery as well. And the amount of indolacetic acid you see circulating in the blood and that you find in urine isn't from the brain. Now, there have been studies that have shown that you can measure indolacetic acid in the brain, in CSF or in perfusates, and that actually represents that tryptamine pathway, because the indolacetic acid in the brain predominantly comes from tryptamine, but of course it can also come from DMT. So the difficulty in establishing analytical methodology and trying to study this in peripheral studies has been that if DMT is very difficult to measure in the periphery, particularly if it's being measured in the central nervous system, being produced in the CNS. And you can measure these other metabolites, but teasing out the other sources for some of these compounds has complicated that. Now, this is somewhat different from my review of some 69 different studies that I did in I think it was 2012. May have been, yeah, where the studies that had found DMT in blood and in urine had reported in some cases very high levels. Right? But my review of those, looking at the technology being used and whether or not the methods were proper for doing what they were claiming they achieved, and whether the methods were validated and proper standards and all that were done, using the standard we use today for identification, a lot of those studies didn't stand up to the test. So in some cases, there may have been misidentifications in those earlier studies, but some of them may have been correct, particularly ones that did find somewhat lower levels. But a whole new sets of studies are beginning now using the more stringent requirements and technology that's giving us a little more insight into what's going on there.
Super interesting. On that note, walk us through the leap from the radioactive tracer work that you did to your 20, I don't know, 12 or 13 LC-MS methodology, where you worked with Rick Strassman and Jim Borjigin.
Borjigin, yeah.
Borjigin. What did that technology finally allow you to prove about the pineal gland?
Yeah. Well, the early work on DMT was being done by what looked like very antiquated methods of analysis today, but they were what we had available. So when I started doing this work in 1976, we were using thin layer chromatography, right? Just spots on a plate that moved with a solvent against the surface of the material on the plate, using different sprays to get the color of the compound, and using radio-labeled compound, mainly compounds, mainly tritium-labeled compounds, so that we could count the number of disintegrations from the radio label to estimate what amount of the compound was present, and of course use radio detection to determine where the compound was on a thin layer plate, right? Well, that moved on to GC, gas chromatography, with various different detectors, converting these compounds into derivatives that gave better signals on the particular detectors, like electron capture detectors or nickel 63 detectors that could see transitions.
And you could do 24 compounds at the same time, right? That was the
Now,
advantage.
this was before we got there. We were doing maybe 3 or 4 or 5 compounds at a time. And that evolved into doing GCMS, which gave us more sensitivity and specificity. And then came liquid chromatography, mass spectrometry, which was the greatest advance in being able to do these types of analyses in terms of biological sample analyses in general. You can now keep the sample in a liquid matrix, inject it onto a column, have high resolution separation of components, and get the exact mass spectrum of it. So using the criteria of one, that it's extracted, two, that it's separated on a column and has the right position where it should come out on a column based on reference standards, and that it had the right mass spectrum and fragmentation pattern, you have unequivocal evidence that is both scientifically certain, but would also stand up in the court of law. I mean, it's, that kind of certainty. So, with those steps, we were able to start applying it to liquid samples. Well, Rick had contacted me, I think back in 2002. He still had some of his samples from his patients where he had administered DMT in, I believe, in 1993, 94. And he wanted to look for other metabolites in those samples to see whether or not there was any better correlation of metabolites with the effects the patients had reported. We got the samples, and unfortunately they were a little bit too old, and we really couldn't get any further data, but we started up a relationship, collaboration, and eventually friendship. And of course, he invited me to participate in his documentary, and we exchanged a lot of information and ideas on things. And he introduced me to a lot of different collaborators, including the work we were doing on ayahuasca, and to Strassman Borjigin, who was at the University of Michigan. Right?
Yeah.
And he told me before he introduced me that they were running rat brain perfusates. They were inserting probes into certain areas of rat brain and collecting the perfusate from that using artificial CSF, and that they were running it on another type of system to try to identify the compounds. They were routinely measuring things like epinephrine, norepinephrine, serotonin, and other neurotransmitters using that system, but that they kept seeing this lump in their chromatogram that was further out, and they wanted to know what it was. And of course, I had advanced mass spectrometry available to me in my laboratory, and said, sure. And we have liquid chromatography, so we can probably take these perfusates and just directly inject them without having to extract them and see what we could find, and we'll go hunting. And of course, he was particularly interested in whether or not we could find DMT or 5-methoxy-DMT. And I looked at the chromatograms and the mobile phase that they were using, and based just simply on the polarities of that environment, the solvents being used and the type of column being used and the relative retention time of known compounds, I felt there was a high probability that this could be one of those compounds. So I had my technician that was running those instruments at the time do the analysis without me ever looking at it. I wanted her to reduce the data, to go through it and determine if any of these compounds, this list of 24 that we had come up with, 24 or 25, were present, right? And she came back to me and she said, yes, said we found serotonin, we found tryptamine, we found the melatonin, and we found the precursor for melatonin, and we also found DMT. Okay. So I looked at the data, and I think I had a smile on my face for about 2 weeks, right? And the data was as good as you could ask for. It was right retention time, right molecular weight, right fragmentation pattern, right ratio of ions. It was unequivocal identification of DMT in rat brain, right? Now, coupled with the CSF from human work that had been done years earlier, right? It's starting to look pretty good, right? So she had a graduate student in her laboratory that was working on these profiles of neurotransmitters in perfused rat brain. And I sent him a standard for DMT, and he ran it, and lo and behold, it matched, right, as far as retention time on his system. So they started another program looking in greater detail at this compound, and that resulted in another paper. We published our initial work in 2013, the one that you mentioned. And I was the lead author, and it was all the work was done in my lab. The samples came from Dr. Borjigin, and of course Rick had introduced me, so he's on the paper as well. So that study was the first to identify the hallucinogen dimethyltryptamine in living rat brain, right?
So when she told you, I'm just curious, how long did it take you until you called Rick and Virgin?
Well, it was pretty fast.
Were they excited as you were? They were like, "Oh my God."
Well, yes.
That's
Well,
exciting.
Rick had for some time, he and I had talked about how we could identify it and whether or not it was in pineal. I said, you've made the hypothesis. It's not unreasonable to hypothesize that, given the melatonin is produced there. Um, it could be in other areas of the brain as well. But let's see what we can do. And we tried isolating it from rabbit pineal and from isolating pineal glands, and we weren't having much success. But there's nothing new to me about that. We were trying to isolate DMT from whole brain tissue from rats for years, and the method would more often than not fail. And it's because of the chemical nature of DMT. It's sensitive to light, it's sensitive to air, it's sensitive to monoamine oxidase. You try to block it, but you can't. You try to block it beforehand, before you collect the tissues. And you do all these things, but the recovery of it from whole brain tissue was miserable.
Well,
you know,
we thank you for that work, basically proving unequivocally, like you said,
yeah,
that the body produces it. If it's pineal gland, I think now we know it's lungs, liver, adrenals. Um, but now I think we can, let that go. And incredible how you did that, to be honest
yeah,
with you. And your persistence and the collaborations that you have is incredible. But now let's shift more towards what does DMT actually do in the brain when it
yeah,
activates serotonin 2A receptors? What's happening neurologically in those moments when people report contact, for example, with non-human entities or profound shifts in consciousness?
yeah,
Can you talk about that?
sure. Well, the first thing you have to keep in mind is that if DMT is a naturally occurring neurotransmitter in the brain, the levels of it are quite low, magnified by going into the synaptic cleft, where the concentrations in that volume can become very high. It's probably distributed in specific brain areas. It's not necessarily everywhere. That if you administer a bolus of that psychedelic from the periphery, and that goes through the bloodstream and is taken up quite rapidly into the brain, basically what you're creating is an overdose, right? So DMT is now going to brain areas where it may not naturally occur. It's going to brain areas where it does naturally occur and has many more receptors, but they're being overwhelmed. The natural interaction with other neurotransmitters and other molecules at the neuron is disrupted, right? And what you get from this is this psychedelic state, right? So we don't normally walk around hallucinating. Well, not all of us anyway. And, the psychedelic state is a unique state that is created, as far as I'm hypothesizing, is created from overwhelming a naturally occurring psychedelic neurotransmitter system within the brain. And that's what I covered in my 1981 review, and postulated that there was a endogenous hallucinogen neuronal system using DMT as a neurotransmitter. So in that process, you experience all these rushes of images in particular. There's some synesthesia that's also involved, where different areas of the brain for sight now are activated by sound, and vice versa, and all of that. And so what is going on there with the overdose? Well, it's overstimulating the naturally occurring sites. It's creating stimulation in perhaps some other sites by overwhelming other neurotransmitters and their ability to bind at their receptors. And it's doing something else that we did not realize until the last 10 years, right? It's stimulating dendritic growth. It's stimulating new interneuronal connections. It's carrying out neuronal repair, right? So in stimulating those that neuronal growth and stimulating the new connections, we have the brain now interacting with perhaps with areas of the brain it doesn't normally interact with. And the latest studies have shown that it actually creates a hallucinogen patterning of the brain, that certain areas of the brain are now interconnecting with much stronger interconnections than they normally have, and that they are making new interconnections with areas of the brain that they don't normally interconnect with, right? So we're reshaping, reforming the function of the brain at the time we're doing that, while stimulating all this dendritic growth, right? So, signals being sent through those new interneuron connections are creating these images, right? The areas of the brain that are processing all that is now quite different, right? And probably highly variable in terms of shifting from one thing to another. And you get all of this that is part of the DMT space, as people are calling it. So, but what does it normally do? It's normally it appears to be involved based on new technology and new results. It's involved in neuroprotection, neuronal repair, response to hypoxia, as well as helping modulate the immune system, and a variety of other things are being discovered. It turns out to be a quite potent therapeutic in its ability to also act as a strong antioxidant and to slow the deterioration of nerve cells that can occur from stress. Right? So, it appears to be that neurotransmitter that's been missing, in our understanding of how the brain works and maintains itself and responds. It's like we didn't understand how opiates really worked until we discovered the opiate receptors. Well, until they were discovered by Saul Snyder and his other colleagues, Candace Pert in particular. We were trying to explain how the opiates work based on the neurotransmitters we knew we had. epinephrine, norepinephrine, dopamine, GABA, all of those at one time or another were offered as an explanation for how opiates worked. Right? The same thing with the psychedelics. We've been saying, okay, it's serotonin, it's serotonin, it's serotonin, it's dopamine, it's serotonin and dopamine, it's serotonin, dopamine, and GABA, right? It's all the neurotransmitters that we know about. Well, what if there's already a neurotransmitter there that is a known psychedelic that is involved in these things that we've described for their natural function, and that we're overwhelming that system and creating these things we call hallucinations. Right? And if you've got a system like that, if it is a neurotransmitter, then you can have alterations in its activity that are normal and abnormal in disease states. You can produce too much, or you cannot produce enough, right? And everything in between. So, some of the effects of these psychedelics on things like depression, PTSD, and a variety of other disorders that's being
Addiction
looked at
perhaps.
now.
Yeah.
Yeah. That are being affected by the administration of a psychedelic, including DMT. When you administer DMT to some intractable people that are suffering from intractable depression, they get not only immediate relief, but prolonged relief, from that effect, right? It's kind of like the Awakening, Oliver Sacks, book on giving people L-Dopa that had, rigidity and Parkinsonism, right? And they were suddenly able to move, right? Well, same thing here, given that neurotransmitter, these people are relieved of the symptomology, but in a way cured. Right? There have been people that have claimed in some of the clinical trials data that is coming out that it has cured some of these people of their PTSD, of alcoholism, of other types of behaviors and mental disorders, but depression in particular. Right? So, and in disorders where maybe you produce too much or the neuronal connections are altered so that DMT related neurons are interconnecting with areas where normally we don't have those types of interconnections, right? And you're getting mental disorders like psychoses and others that have hallucinations as symptomology. I mean, it just opens up a whole new world of pharmacology as well as understanding of this thing we call consciousness.
Well, two interesting things come to mind. And would you go as far as to say that, we know that sleep is important, but then we know it's not the quantity, it's the quality of sleep, right? And in quality, we mean how deep, what stage are we in? And you have to be in a pretty deep stage to achieve REM sleep, right? Rapid eye movement, which in it by itself may be hallucination, right? So would you say that that is really DMT's action that achieves that quality of sleep, which then leads to, I don't know, activation of the glymphatic system and the neuronal plasticity and health?
Yeah, the purpose of sleep is something that has been researched for a very long time, but it seems that the most important component of that function is that it helps clean the brain.
Mm-hmm.
Right? So the interstitium, the spaces between cells and the glymphatic system, but the more newly defined system of the interstitium, helps remove a lot of these compounds, and at the same time, there is neuronal repair and new connections made and old connections snipped, right? The brain is not completely remodeling, but it is undergoing a what's known as a plastogenic effect, right?
And I think part of that was when I read about the glymphatic system, and I think it's fairly young, 15 years maybe, I think, if I'm
Mm-hmm.
not mistaken, that one of the core drivers of that is actually that the brain is switching into a different pulse frequency and synchronizing its propulsion of toxins and waste products from the glymphatic. I think
Yeah.
it's the thoracic duct that it ultimately drains in.
Mm-hmm.
Uh, but that's really incredible. Now, on the other side, um, in the mental disorders that you mentioned, uh, I think it's the truth when you say that schizophrenic patients have a lower rate of getting cancer than the regular population. Um, that they actually live a long life, even though it's potentially miserable because of all they have to endure mentally. But what are your thoughts on that? I mean, there would be kind of from both sides, you could say, all right, well, that leads to better health, REM sleep, maybe because of DMT, and I guess a prolonged life because, even if you have schizophrenia. But what was it that there are very rarely reported cases of smokers and schizophrenic, and 80% of them smoke or something like that?
Yeah.
I don't know. I mean, I don't want to
Well,
sensationalize this, but
there was a new paper that came out in 25 about the relationship between psychedelics and the immune system, and it showed that these compounds are potent anti-inflammatory compounds through the effect of these compounds on serotonin. They initiate a lot of anti-inflammatory effects and modulate the immune system. Um, and this may be part of the ability to smoke and not really get disease from it or from living longer life, is that their inflammation levels may be much lower. I haven't looked at the research on that, but I certainly will. Right? So the psychedelic control of that system was published in a paper in 1925 by Chung in laboratory of Michael Wheeler, right? And they addressed any number of possible roles for this compound, for DMT or for other psychedelics, and being able to modify that system, both the anti-inflammatory effect and the communication between the brain and the immune system, enhancing brain crosstalk, that they also are broad immunosuppressants, but they are more specific than a lot of the immunosuppressant drugs that we're using today, right? And they believe that these compounds may be used as targeted immunomodulators, right? And the most exciting thing that has come out of this psychedelic research is the development of new pharmaceuticals that can have the uh, effect of uh, enhancing plastogenesis, right, and effective in treating perhaps depression and a range of other disorders that the psychedelics have been shown,
without the
but
hallucinogenic effects.
they're
Yeah.
not hallucinogenic.
Yeah. I think
So
David Olson, that's his work. He
yeah,
he works on that with
yeah, there have been a number of exciting studies that have come out on that. They actually have given these compounds their own class and name. They're called PIPPs, right? And that means they're psychedelic informed, but psychedelic experience inactive.
wow,
PIPPs, right?
hippies.
These are all new, and some of them are currently undergoing clinical trials.
I think David calls them psychoplastogens or something.
Yeah, yeah.
Yeah. Wow, that's super exciting. I mean, we
Hmm.
need to fund that type of research for sure. So here's to Bobby Kennedy. Um, so there's a hypothesis emerging.
Hmm.
Now, I initially I contacted Michael Angelo at Stanford with Diane, and we were proposing this hypothesis that it's produced perhaps in autism in excess, and
Hmm.
threw around the idea, and he's like, "Well, that sounds amazing, but we need a person that could, write the essay for us and find out about the chemistry and all of that." So initially, I thought of Rick, and I contacted him. I read his book in the 90s, I think, DMT, the spirit molecule, and I was always fascinated with that topic, and listen to guys like Joe Rogan, and there's more papers by others as well. But he quickly, sent me your way, and
Yes.
that's how we got in touch with it. But with you, and you so much helped us to eventually, introduce us to David Olson, and wrote the methodology of our study that we're doing. But anyway, um, you mentioned schizophrenia. I proposed autism because of the work that I've been doing, and pretty much uh, a lot of it is so congruent with what you're saying in regards to the sensory system, um, in regards to a lot of the symptoms that might be emerging if you hyperactivate these receptor systems, right? Especially with the motor discoordination, the apraxia that we see, and but in regards to the function and the overstimulation of these systems that you mentioned, what could you expect in a chronic low dose, elevations of these tryptamines? What could you expect? Maybe, it's too far out to hypothesize that in a developing brain,
Hmm.
but would it be chronic activation of these receptors, or would there be some sort of tolerance that develops and actually a downregulation of these receptors? Because from what I know is that psilocybin, you can't take that every day.
Right.
You should take breaks. But with DMT, there's a consistent effect, even if you would do that every day. Although I did hear of some reports where for some people it just stops working for 2 months even. It's almost like its own hierarchy or empire that lets you in when it wants to. But can you talk about that? Like, is that even plausible to come up with a hypothesis like this, that it could be elevated in autism? Because the other thing is, you said it's the neuroplasticity. We don't really see any damage in the MRIs of autistic children, teens, adults, even in the 40s. We don't see brain damage, despite everybody talking about, oh my God, it's neuroinflammation and all of that. So, in fact, we do see hyperconnectivity in certain regions in the brain that do have synesthesia, like you mentioned, those that report it by typing. So, what are your thoughts on that?
Yeah, I think that's one of the real possibilities, given some of the histopathology that has been observed in having extra uh, and overgrowth of connections in neurons, and some of the symptomology that you've described and that I've read about, of being perhaps so overstimulated they can't talk. Right? There's a world going on, so rapidly in their brain, that they're so in their head, that their affect and everything is what it appears as one of the symptoms. Um, and certainly, that would be something that would be able to be measured. Um, the DMT that has been identified and consistently um, confirmed to be present in tissues has been in the interstitium. Right? We got it from perfusates, we got it from cerebral spinal fluid, um, perhaps going into the glymphatic system, looking at saliva and a variety of other things that are derived from that same process. We might be able to start measuring this. Unfortunately, of course, we can't do brain perfusions in these kids, and get a definitive answer. But again, the fact that you don't see brain damage as such, and you do see these hyperconnections, certainly meets in the symptomology that is seen with what is now the known pharmacology of this neurotransmitter, right, of DMT. And like I said, it may be the reason we haven't found an answer to it is this has been one of the missing neurotransmitters that we haven't sufficiently studied or addressed in terms of its ability to inform us about the causes of some of these different disorders. So certainly starting those kinds of measurements by looking at things that would be related to levels that we could measure coming from the brain, looking at the interstitium and glymphatic system, and being able to determine what's going on in the gut, become very important. Because if this is something that begins at birth, and another thing that has come up from all this is the probability that DMT is involved in brain development, in neurodevelopment in children, that if that goes wrong in any particular way, you would see the wrong connections being made, too many connections being made, not enough in some areas, too many in others, that could produce this whole plethora of symptomology, right, for different disorders. And, finding something that could address what these kids have to go through through life is something I would be very interested in addressing. I mean, if it is possible that this is involved in this particular disorder as well, then guns ho.
Yeah.
Yeah,
I
let's
mean,
go.
do people usually talk a lot when they have DMT, uh, exogenous experience?
Well, I never did. No,
Me neither, not yet.
yeah, I can only really talk about my own experience, but you're so involved in what is going on in your head,
Mhm.
right? The places that it appears to take you, right? And where you arrive, it's
It's not necessarily a thing to take if you want to socialize with your friends.
not really. It's not a socializing drug,
Right.
no, because my guess is the world you're experiencing with somebody next to you that also took it, you're not in the same place. Yeah. But that,
Wow.
that kind of goes to that whole idea of entities, right? These worlds that are created in our head from taking psychedelics. The
Are they created by our head, or are we just fine-tuning to tune into the bigger consciousness to see what's.
well, I'll tell you what I think about that. Our first experience of an altered state in another world is dreaming, right? Now we get there without having to take a psychedelic or a hallucinogen, probably because we already have one, right? And that's what it is doing. At night while it's cleaning the brain and helping do repair and everything, it starts making these connections and stimulating neuronal growth, and we have dreams. Same thing may be true for creativity and imagination, and certainly it may be involved in fantasia. There are people that are aphantastic that can't bring up a mental image, right? And there's some that are hyper fantastic and they can visualize, whole worlds, going on around them and actually have interaction with it, right? But it's normal, right? We all can more or less, bring images to mind, right? So one of the issues with psychedelic research, of course, is set and setting and expectation. And it was Terence McKenna, the one of the scientific gurus and of psychedelic chemistry and medicine that came up with the idea of machine elves. And son of a gun, now that everybody takes DMT, they're looking for machine elves, right? Now, why is that? It's expectation. The uh, more primitive tribes, I don't think anything's primitive anymore, but the use of hallucinogens in culture in tribal communities and Aboriginal communities, um, they didn't report machines. They didn't know what machines were. They reported their environment. They would see predators and fantastic beings and all of that, right? So I think machines and all that in our hallucinations are uh, construction that we make, in our heads. And the same thing with dreams. I mean, we don't need hallucinations for in many cases. I mean, don't need hallucinogenics for some cases. Fantastic dreams and visions and all of that. But again, it may be because we've already got it in our heads. So I'm afraid that the entity thing has gotten a little bit out of hand to where we're trying to get knowledge from superior beings. Now, what does that sound like? The start of a religion.
Yeah, I'd be very careful with that too.
Yeah, I think we have to be very careful with that.
And
Uh,
that's not what we're here to answer.
well, not really,
You
but
know, if
people
we
wonder.
even if we find this to be elevated in autism, it's how can we quickly reverse this is my goal. I not that I disrespect anybody with autism. There's going to be people offended when you say, oh, I want to fix something. It is who I am. But yeah, not for the non-speaking autistic child that's banging his head at 2 in the morning. It's not. I'm
Mhm.
sorry. And those are the ones that we are studying. Severe nonverbal autism. What is it? The age between age 10 and 30, and we're going to start with 10. We're going to have a control group of 10 that are age matched and sex matched. And we're just gonna see what we find.
Sure.
Urine, blood, saliva, why not? And then maybe apply it to different conditions and see what we find. Maybe it's low in depression, maybe it's high in schizophrenia, maybe it's low in Alzheimer's. We'll see. But
Mhm.
we're just here to set the record straight and maybe start a new chapter in neuropsychiatry. I mean, what do you think? What could be the impact if we find that we can not only measure these more easily, these compounds, but um, maybe affect treatment modalities.
Yeah, well, the new pharmacology that has been discovered for these compounds is already leading to new therapeutic targets and new therapeutic drugs that will alleviate a lot of symptomology that's less than pleasant. The same thing with autism. If it is an ongoing overstimulation, an ongoing plastogenic effect from having too much of this, which is, I would hypothesize that it'd be too much, not necessarily enough, and that the binding sites for these psychedelics are well defined, that drugs can be designed to treat that, to block that binding site or to prevent the increased synthesis, enhance the turnover. So the David Olson's lab has produced a lot of fabulous work in this area, including defining a different receptor for the binding of psychedelics that appears to answer the question of how do psychedelics produce the effects that they do. And this turned out to be a serotonin receptor, but it's inside
Sigma 1.
neuron, right? It's not the ones that we've been studying on the cell surface, right? So the serotonin receptor on the cell surface, of course, interacts with DMT. That's probably part of its behavior and role, but certainly it binds serotonin, right? And serotonin has a lot of effects. But serotonin, unlike DMT, cannot get inside the cell. So if DMT, which can get inside the cell because it's very lipophilic, and bind to the internal 5-HT2A receptor, then that's not actually a serotonin receptor. It's the DMT receptor,
Yeah.
right? It's the psychedelic
Yeah,
receptor.
I think that's the internal machinery. I think it's connected to either the nucleus or the mitochondria and activating these calcium channels that
Yes.
then kind of power this arboration of the synapse, to
Mhm.
reach out to the other
Yeah.
synapses. It's almost like starting the motor within the cell.
Yeah.
And yeah, it's interesting because, for autism, remember there are only two FDA approved medications, right? Which is risperidone and Abilify. And these are antipsychotics, and lo and behold, they block out of the 300, I think, receptor systems that we have in the brain. Oh, they block 5-HT2A. What a
Yes.
coincidence. And that would make
About that.
sense why these medications not work because of what you just explained, because these medications don't follow inside of the cell. And
Yeah, exactly. And that needs to, be looked at as soon as possible because this receptor inside is mainly associated with the Golgi apparatus.
oh, the Golgi, yeah,
Yes, yes, it's the Golgi
that's
that
what
is responsible for packaging all the proteins
that's right.
that go out to do all the wonderful things that neurons do, so that it can, stimulate new growth of dendrites and make new connections and extend the axon and do all of these things. So, how it's regulating all of that traffic, it goes on inside the cell as well, is very important and could be being overstimulated.
Yeah, I wonder. About 8 years ago, when I did one of the tests that I recommended for parents was this urinary neurotransmitter test, and we did, I don't know, over 100 of them, right, in autism. And we measured everything from dopamine, serotonin to glutamine, histamine, and the precursors and the breakdown products. There were like over 20 markers, and it was fairly cheap. And the one thing that consistently came back high was tryptamine. Serotonin was variable, but tryptamine was always high. And back then I was like, that's not important. It's just a whatever precursor metabolite. I didn't question it. But then when I started my work with my colleague Alex Saharikis with the testing the microbiome, and we've tested now whatever over 3,000 stool samples, really with the 16S, sequencing, have a great idea about the microbiome. What most other stool tests don't test is these tiny elevations, of these bacteria that have the TDC enzyme in it. And if they're overgrown, then they might suck up that dietary tryptophan to funnel towards tryptamine, and that might be the reason. Because one of the things that Michael at Stanford, when initially one of the first things he said, you know what, that's a great hypothesis, but there's no chance that the microbiome synthesized uh, DMT or tryptamine would ever reach the brain because of MAO. And that's when we were like, well, wait a moment. We also did a lot of genetic testing, and we saw that, first of all, boys, the MAOs, it's X-linked, so they only have one. It's a hemizygous, if that gene is affected. So that would kind of explain why boys are affected more than girls if they have a polymorphism in that MAO gene, and
Right.
most of them did. Um, and they had a slow working copy, and okay, that's when he said, all right, call your contacts. And that's when I contacted Rick, and he sent me to you. And I was actually very surprised that you said, hey, let's do it. I thought you would have said, no, that's biologically impossible. There's no chance. I've done this for 40 years. There's
Yeah. Well, the other part of that is these compounds don't have to go to the brain to have an effect. I mean, they can be produced in the gut and use the second brain, the enteric system. right?
You mean the vagus nerve, perhaps?
Yeah, the vagus nerve to send signals to the brain, and depending on how that's connected,
I
you
never
know,
thought of that. That's
yeah,
incredible. Wow.
yeah.
Wow,
We've
wow,
got
wow.
we've got more than one brain, so it's
Yeah, you're right. I mean, this is very interesting. Okay. Anyway, I don't want to stretch this out too long. I think we covered a lot of ground. Super, super interesting. I know you're coming out with a book. Can you tell us about that? I think it's called Reality, you mentioned.
it's reality shadow, which comes from a quote by Blake that this vegetable universe is just reality shadow, right? And what it's about is about hallucinations, and more specifically, the hallucinogen DMT, and the implications of its discovery as naturally occurring in brain. So it gives some of this history that we've discussed, today, and how psychiatry has evolved, from looking for these compounds, and the new pharmacology and what it says not only about our neurophysiology, neuropharmacology, and neurochemistry, but things like consciousness and perception, creativity, imagination, dream states, everything that we constitute as a hallucination. And it redefines hallucinations as puts it on a molecular level instead of a more psychological level.
When is it going to be released, Steve?
Well, I'm just finishing it now. I'm probably going to self-publish because it's a much faster way to get information out there nowadays. And I did it backwards. I didn't send out a proposal when I first started working on this. I just went ahead and wrote it and finished it.
Nice.
So fairly soon.
Well, let us know. We certainly help you get the word around. Um, do you think we're on the right track, Steve?
Yeah, I do. Being in the field for 50 years, I mean, if you outlive everybody else that was in the field, you become the expert, you know. So right now, uh, you know, I, I I have been able to assimilate an awful lot of information in this area, and I really appreciate people contacting me to get my insights or opinions. Uh, and the more I look at it, and the more I look at what could be coincidence, but when I pile coincidence upon coincidence upon coincidence, and uh, factual scientific data added to that, I definitely believe that DMT has been a missing neurotransmitter, and that our understanding of why it's there and what it's doing will be of great help to humankind.
My last question,
Sure.
and I thought of that very carefully. When Cassie will call you from UC Davis saying that she found elevations of DMT in autism, will your smile last longer than 2 weeks?
Well, as they're slowly slipping me into the crematorium, I'll probably still be grinning.
Nice. All right. Well, listen, we couldn't do it without you. You've been pushing this forward.
Thank you,
I'm
Christian.
really thankful. I'm excited to see what's on the horizon. It's an honor to be working with you on this project, and we'll keep everyone up to date. Of course, it would be amazing if we could jump back on. So once the results are in, that you make sense out of them and tell us about the validity and techniques that were used with Cassie and David at UC Davis. So with that being said, anything else you want to shout to the world, Steve?
Watch for the outcoming book.
Yes. Reality Shadow. Yeah.
Yeah, reality shadow, and
Amazing. Well,
keep an open mind. Just keep an open mind.
absolutely, always as a scientist. So, all right. Well, all the best, and we'll be in touch, Steve. And thank you, everyone, for listening. Hopefully, this was helpful. I think it was amazing. My mind is blown, so I have to digest some of the info that you mentioned. But yeah, vagus nerve. That's what I'm going to look up next. So, all right. Well, have a great day, Steve.
You too. Thank you very much, Christian.